All right, part 2, cell cycle. Please do what you were supposed to do for the other materials in class. Make sure you've signed into AP Classroom to get that stuff finished. Okay, describe the basic stages of the cell cycle using the diagram provided. So this is your basic cell cycle diagram here at the bottom. We know that most of the cell's life spend their life in a phase called interphase, and only a short period of the cell's lifespan is actually going through mitosis or cell division. The largest phase of interphase is G1, where the cell is growing, developing, gathering resources, all of these things. And then we know that when the cell reaches a certain size, right, the surface area to volume ratio reaches a certain amount, then the overall efficiency of the cell will drop. So at that point, once it reaches a certain size, it needs to consider dividing into a new cell. So G1 then enters into S phase of interphase. S stands for synthesis. Synthesis means making something. What are they making? Well, that's the stage where it's copying the DNA. Gets all copied. All the genetic material gets copied so that all of that can be placed into a new cell. Remember, we want to have any new cell we make have exactly the same genetic information as the other cell, because if it has errors or incomplete genetic information, then that's how diseases creep up. The last stage is the G2 phase. G2 refers to kind of final kind of growth and preparations for cell division as well. And it enters into mitosis. And then a stage that's not really considered mitosis is cytokinesis, right? Kinesis, kinetic, from the Greek word for movement, and cyto for the cytosol is moving. That's when the cytosol and rest of the cellular components get separated into two separate cells. And then, of course, it's a cycle, so then it starts all over again. The two daughter cells that are produced then enter into interphase and going through G1, S, G2, mitosis again. Now, remember that in not all cases, cells go through this process as well. For a lot of the cells in your body, right, they'll go through G1, the cells will reach a certain size, and instead of skipping over into S phase, they'll go through a period called like cellular quiescence then, which means that they just kind of stay the same. So these are things like your nervous system cells and to an extent your liver cells, right? They enter into what's called the G0 phase, where they just kind of stay. And so kind of a holy grail of cellular research right now is how can we trick some of those cells that have kind of stopped and they're stopped and stuck into that G0 phase, can we trick them into going into S phase of interphase and so that they can divide? So that would be a great thing, say, for Alzheimer's and areas where there's a lot of brain damage, so that cells in the brain can regrow and correct damage in the brain, just like cells in your skin divide and correct any damage in your skin. But for now, trying to trick those cells into going into S phase has proven to be difficult. The next question says, what would result from a cell that completed the cell cycle but not undergo cytokinesis? So this actually does happen. It happens in muscle cells in your body. If it didn't undergo cytokinesis, it would not divide into two separate cells, but you would have two separate nuclei within the same kind of cell. And these become what are called multinucleate cells, meaning that they have more than one nucleus inside a cell. And these are muscle cells are like this. And so when we look at muscle cells are very long and thin cells, and that this is a way that they can kind of increase their efficiency considering their larger size, because they have a second kind of engine that's producing materials that they need to do the work of the cell. Okay, identify the 5 major stages of interphase, of mitosis. So of course the first stage is interphase. And interphase is determined by the fact that the nucleus and the nuclear envelope, these are all very distinct. You cannot see the chromosomes at this point. You cannot really see the DNA. At this point, the DNA is tightly wound into very small packages called chromatin at this point. During prophase, though, that's after the information has the DNA has replicated, the chromosomes then appear. Remember that the word chromosome just means, chromo in Greek means color, and somo means body. These are colored bodies referring to the stain that chromosomes can get during prophase. So these are colored bodies that appear only during prophase. Okay. And then during prophase, right, you can see the chromosomes at that point. They appear, they're very distinct, and the nuclear envelope begins to break down as well. During metaphase, right, the chromosomes then line up in the equator of the cell, what's called that metaphase plate, and they're attached to the different spindle fibers. Remember that animal cells have structures called centrosomes, and centrosomes are built of centrioles and microtubules. So they help organize the movement of the chromosomes to the opposite poles of the cell during cellular division. And then the next phase, anaphase, the chromosomes start pulling apart and they start moving towards the centrosomes or the centrioles on the opposite poles. And then telophase, the chromosomes are now at each pole of the cell, and they're starting to kind of break down back into chromatin again, and the nuclear envelope is beginning to reform again. And then the final stage is we have cytokinesis, right, where the cytoplasm and everything kind of pinches off, and we have two cells. So you should be able to, in a way, kind of draw, label, describe that whole process. Okay, the role of motor proteins in mitosis. Remember that in some ways we can view the cell as a lot of things just kind of floating around and diffusing from areas of high concentration to areas of low concentration. But in other ways, how are things organized and how do proteins move along? Remember that the cytoskeleton actually makes a very kind of complicated series of kind of highways and roads throughout the whole cell. And it's very interesting that things like motor proteins kind of attach to the cytoskeleton of the cell and help pull things along to move things into different orientations. So if we think about it, right, so you've got once your homologous chromosomes replicate, the homologous chromosome being the chromosome that you get from your mother and your father, and once they replicate and duplicate, right, then you've got what are called the two pairs of sister chromatids. And those are always kind of complicated, trying to get the vocabulary correct between chromatin, chromosome, homologous chromosome, and sister chromatid. You need to spend some time with that vocabulary and make sure that you've got them kind of nailed down so you know exactly what you're talking about. But each of those homologous chromosomes, right, gets duplicated and they get attached in the center by something called the kinetochore. And the kinetochore is a series of proteins with attached microtubules, which then attach to the centrosomes in the two polar regions of the cell. What the motor proteins then do is they attach to the kinetochore and they help kind of, through air quotes, kind of walk the chromosome along the microtubule to the opposite pole. Now, as it's kind of walking and pulling that chromosome to the opposite side, that microtubule is being fed out through the other side of kinetochore, and it's actually being depolymerized. So it's breaking down into its components, right? This, when it was first discovered, became known as kind of the Pac-Man mechanism because it looked like kind of Pac-Man eating its different pieces. So those are the roles of motor proteins. How is cytokinesis different from mitosis? So mitosis really just refers to kind of the duplicating of genetic material and getting it from one pole of the cell to the other. Cytokinesis actually refers to the actual division of the cytoplasm. It happens a little bit differently whether you're dealing with plants or animals. In animals, cytokinesis occurs during cleavage. So kind of if you look at it closely, right, you get that kind of cleavage furrow of the cell. And then along where it's actually cleaving in the center, there's actually a ring of actin microfilaments that go around the cleavage filament. And it acts kind of like a little rope or a belt that tightens throughout time. Okay? And as it tightens off, it's kind of a contractile tightening ring, it kind of ends up pinching off the cell into the two daughter cells. Now, plants don't form cleavage furrows. What happens is that vesicles from the Golgi apparatus move to the middle of the cell, and they kind of line up. And what they kind of coalesce, and they form what is called a cell plate. And the cell plate will eventually turn into the new cell wall. So remember that animal cells are very kind of flexible and loose because they only have that kind of plasma membrane, whereas plant cells tend to be more rigid because they need to maintain they don't have all those kind of structural mechanisms to maintain their shape, so they've got those strong cell walls. And the cell plate eventually becomes the cell wall. Famous experiment here. So the question is, how are all the different stages kind of regulated within the cell cycle? So how does the cell know to move through the different phases of interphase and into mitosis, finally into cytokinesis? How does the cell know how to do this? Well, like most things through cells, right, these things are all controlled through signaling molecules. Okay? This is very similar to other forms of cellular communication that we already talked about. And so to kind of prove this in a way, or to demonstrate this idea, right, they took two cells here in experiment one cell that was going through S phase, so it was duplicating its chromosome, and one phase into regular kind of G1 phase. So what they did was they took components from the cell, kind of the cytoplasm from the S phase of that cell, and they injected it into the other cell that was in G1 phase. And what happened was that it triggered the G1 cell to enter into S phase. Likewise, they tried a different thing. Well, G1 and S phase are very close together. Let's do something that's kind of farther apart. So let's go from something that's in mitosis, M phase, and a cell that's in G1. Those are kind of distant, kind of far apart throughout the cell cycle. Okay, let's extract cytosol from the M phase cell and let's inject it into the cell G1. And let's see what happens. Well, just like the first experiment, the G1 cell then entered into M phase. It skipped S phase and entered right into M phase. I don't know what that would look like. So there's obviously some sort of signaling molecule or something that's present during one stage of the cell cycle that can trigger cells to enter into different stages of the cell cycle. Identify and describe the three major checkpoints of the cell cycle. Okay, so a checkpoint in the cell cycle is a spot in the cell cycle where these kind of stop and go signals are there. So, right, with G1, it needs to have reached a certain size and collected enough material so that it's ready to duplicate its DNA. If it's not ready yet, then it shouldn't be triggered to go into S phase, because this is how damage and things happen. So there needs to be stop and go signals at these different spots. So there's 3 kind of checkpoints where these stop and go signals can happen. The G1 phase, the G2 phase, and M phase, the 3 checkpoints. Okay, so what are these regulatory molecules that regulate the cell cycle, that provide these different signals? Well, what is one of the major cell communication molecules that we know of? Well, I hope that your mind goes back to a kinase and through phosphorylation events, because that's what happens. So we've got 2 that are very important. We've got kinases and cyclins. Okay, so CDK is known as a cyclin-dependent kinase. This is your kinase. Cyclin-dependent kinases mean that they kind of phosphorylate cyclin. Okay, so the kinases in a growing cell are considered to be inactive. Okay, they need to be kind of switched on. To become active, the kinase must be attached to a cyclin molecule. Okay, so how does the cell control this? Well, the cell kind of fluctuates the concentration of cyclin molecules throughout its cell cycle. So at one point in the cell cycle, the amount of cyclin is very low, and other parts it gets very high. Now, the amount of CDKs, cyclin-dependent kinases, they also increase and decrease according to this other molecule called MPF. And MPF stands for the maturation promoting factor. So as MPFs increase, the concentration of MPFs increase during the cell, also the amount of cyclin also increase during the cell. And these MPFs, they can also act as a kinase, phosphorylating proteins that help break down the nuclear membrane, and they also assist in chromosome condensation and spindle fiber formation as well. Okay. Explain the role of APCC. This is an important checkpoint during the M phase, and this molecule helps trigger the transition from metaphase to anaphase by targeting specific proteins for degradation. Okay, so if we think about what's happening during that point, right? Metaphase, we've got the sister chromatids all lined up at the metaphase plate. And then during anaphase, those sister chromosomes are going, or sister chromatids are going to be pulled apart to the 2 opposite poles in the cell. And so those parts of the microtubule need to be degraded to be able to pull it along. So this APC protein helps doing this. Later, this is going to help target specific mitotic cyclin proteins as well and help to degrade them. And then once these ones become degraded, this helps serve as a signal to exit mitosis completely as well. Growth factors in the control of the cell cycle. So growth factors, again, are things like proteins that are released by certain cells that stimulate other cells to divide. Okay. For example, there is one called PDGF, which stands for platelet-derived growth factor. Okay. These are needed for the cellular division of cells called fibroblasts. Okay, so platelets, let's go back a step. What are platelets? Well, platelets are components of your blood. They help blood clotting. So you get a cut, and of course you don't bleed to death because your body can stitch that up, heal the kind of cut, make a scabby part over top of it. Okay, what happens is your platelets go there and they form a kind of scab, so you stop bleeding. Good, so you don't get infection into the wound as easily, and you don't bleed to death. So that's what they do. But they also release factors that are going to help start sealing up kind of the hole as well, and going to promote kind of if you got a cut in your skin, the skin cells to start dividing as well. So there needs to be a kind of trigger that says we need to start dividing some cells. So those particular ones, that PDGF, that platelet-derived growth factor, it triggers these cells called fibroblasts. Okay? And fibroblasts, the reason why they're triggered is because they have specialized PDGF receptors on their cell membrane. And if we think back, well, what kind of receptors would those be? Are those a G-coupled protein? Are those a receptor tyrosine kinase? Those are receptor tyrosine kinases. So we should remember what those are like. And then, of course, so the PDGF is an example of a signaling molecule. And then it triggers a signal transduction pathway in the cells to get those certain cells to leave their kind of G1 stage in interphase to enter into S phase, so they can eventually start to divide and assist in blood clotting. So that they can, no, these are more, fibroblasts are cells that help seal the wound. Now, density-dependent inhibition and anchorage dependence are very important. So density-dependent inhibition, density, how many things are there? So crowded cell, so you place a cell on a petri dish with a growth medium. That cell will continue to divide until all the space has been used up, and then they will stop dividing. If you then take that petri dish that's covered with cells, and then you kill a couple of those cells, then the neighboring cells will then start to divide again and fill in that space. Okay? So this is the idea that sort of crowded cells stop dividing. The cells will continue to divide as long as there's enough space for them to do that. Right? In some ways, people are kind of like that too. People, if there's not a lot of space, people really, don't like living in places where there's a lot of space either, these kind of things. Now, the idea of anchorage dependence means that cells to divide usually need to be attached to some sort of substratum, okay? And for either they need to be on something like a biofilm or something if they're a bacteria, but for cells in our body, they need to be attached to the extracellular matrix. Now, why is this important? Now, this is important. Density-dependent inhibition and anchorage dependence are important in a medical context because cancer cells do not demonstrate these two things. That their DNA has been damaged such that they continue to divide no matter if they're crowded or not. So this is how tumors form. You get a mass of cells that keeps growing and dividing and dividing and dividing. It doesn't stop because normally cells would stop dividing. And then this anchorage dependence, we've talked about that in class too, that most cells have this kind of unique stickiness to them. They stick to other cells and they stick to that extracellular matrix. Well, what can be disastrous with cancer cells is that they lose their stickiness. If you've got a tumor that's stuck in one place, the tumor's not so good, but at least, then it can be kind of medically removed. And at that point, it's called a benign tumor. Because you can have surgery, and if the surgeon's pretty good, can remove most of the cancer cells, and you don't have any problem. What the problem with cancer is that they lose this kind of anchorage dependence. And so the tumor can either break off and start floating around the body, or parts of the tumor can break off. That's called metastasis. the tumor can metastasize and parts of it can break off and then free float around the body and attach themselves to other surfaces and start growing tumors throughout the entire body. And that's when you get this kind of out of control cancer growth. And it's not very, these become what are known as malignant tumors and become very difficult to deal with. And you need to start using radiation. You can't really deal with them surgically. Okay? So yeah, cancer cells do not demonstrate density-dependent inhibition or anchorage dependence. Why is that? Well, because there's been some sort of mutation to their genetics, either through some sort of environmental thing or like an environmental stimulus like smoking or some sort of toxin in the environment, or just the fact, unfortunately, if you live long enough, you're going to acquire more and more damage to the DNA in your cells just through the amount of age that you're living. And then, unfortunately, you may acquire the wrong kind of damage that will damage those genes that kind of control things like density-dependent inhibition and anchorage dependence. So it can be both a factor of environmental factors and simply as age to develop cancers as well. I think we just went through that. Why is cancer considered uncontrolled cellular growth? Because the cell cycle is uncontrolled. It continues to move through the cell cycle into continuous mitosis, and you don't have things like density inhibition to kind of stop it as well. So cancer cells, my notes here, cancer cells do not need cell signals that regulate the cell cycle. They go on dividing indefinitely, and in some ways these cells are kind of considered to be immortal as well. They're continually undergoing. So for these cells, a normal cell to be kind of to be changed into a cancer cell, we say it's been transformed through some sort of mutagen in the environment. Now, it's important to note that, so some cells are not dividing, but if we think about something like a skin cell that's usually going through a lot of division, normal cells, what they do is they'll only go through, they're not immortal, not like a cancer cell. They have a limit they have a limit to the number of times that they actually go through cell division. And the number is an average between 20 and 30 times. Why? Because even though they're dividing all these times, errors are creeping up through the replication. And so this is kind of, in a sense, nature's way of trying to cut off a lot of errors from creeping up into your genetics, because if these errors kind of creep up, then it's possible for things like cancer to develop as well. And so that's why we've got this idea of programmed cell death, apoptosis, where after about 20 to 30 cell divisions, the cell undergoes a kind of cellular suicide or cellular programmed cell death, where the cell will kill itself off so that those kind of errors do not reach a kind of critical threshold. But unfortunately, with cancer cells, they never undergo this kind of programmed cell death. They're functionally immortal in the sense that they continue to divide again and again and again. Now, there are two things. We've got things called oncogenes. The root word onco refers to cancer. An oncologist is a doctor who studies or deals with cancer. So oncogenes are genes that deal with cancer. And then there are ones called tumor suppressor genes, which help suppress and fight against cancer. Now, oncogenes are normal genes that have been transformed to become genes that promote cancer. So these are normal genes that have suffered some sort of mutation, either through some sort of environmental reason, through like smoking, or just through age. These are genes that become transformed and promote cancer. So what are some of these things? What are some of these genes that could be oncogenes? So these are genes that promote, say, apoptosis, controlled cell death. If you shut down those genes that stop cells from committing cell suicide, then in a sense they become immortal, and these cells always live for as long as they're receiving nutrients. These are genes that promote continued growth. They don't get shut off, and promote cell division. These are genes that promote like concentrations of cyclins and kinases that promote the cell cycle. So if these things get mutated or changed, then they don't shut off, and they do exactly the opposite of what they're supposed to do. So an oncogene, the definition is a gene, a normal gene that has suffered a mutation, and they're genes that normally promote cell division and apoptosis. These are your oncogenes. Now, tumor suppressor genes, these are normal genes that are intended to slow the process of cell division. They're also genes that are to repair mistakes in DNA, and they're also genes that are involved with apoptosis as well. So they're genes to prevent the development of tumors in the first place. Okay, so if we can repair, remember when we looked at DNA replication, we'll go through this again. There are specific proteins that after the DNA has been replicated, these proteins then go back through the long stretches of copied DNA to check, to make sure that mistakes have not crept into the copying process. Because if mistakes creep in, this is how abnormalities and diseases happen. And if mistakes creep in with these specific genes, tumor suppressor genes, then things like oncogenes can come along as well. Now, two famous genes in oncogenes and tumor suppressor genes are known as the p53 gene and the RAS gene. So p53 is a tumor suppressor gene, and the RAS is known as an oncogene. Okay? And these control a lot of cellular processes that control cell division. Okay? So these ones control things that are involved in tumor genesis. So things like increased cell proliferation, apoptosis, cellular motility, and even angiogenesis. Okay? Because in angiogenesis, this is the development of blood vessels. So tumors, they end up developing their own network of blood vessels because they need nutrients to kind of feed their uncontrolled cell growth. So they produce their own kind of mini arterioles and venules so that they can receive blood so they can continue to grow. Okay, so RAS is an oncogene. What it codes for is an intracellular protein, a protein that is found within the plasma membrane of the cell. Okay, growth factors, right? These are signals from other cells. They activate the RAS protein, and then RAS starts a phosphorylation cascade, which then leads to certain transcription factors. Remember, a transcription factor is going to be a protein that aids in the transcription of DNA. So it's going to open up a certain gene, and the gene is going to become transcribed to produce a segment of messenger RNA. Now, what do these transcription factors actually code for? Well, they code to make the mRNA for proteins that operate cell growth. So these are your CDKs and your cyclins. Okay? So what happens is, if RAS is mutated, because RAS is a protein, it's going to be made through DNA. If that DNA becomes mutated, meaning that there'll be different kinds of amino acids in different spaces, so the RAS gene is not, or the RAS protein is not functioning properly, then what happens is that you get a non-stop phosphorylation cascade. the phosphorylation cascade that's involved in the transduction pathway of this particular gene cannot be shut off. And so you get a non-stop transcription factors, you get non-stop cyclins being produced in the cell, and then you get non-stop carried away cell cycle, going through all the stages of the cell cycle, promoting development of cancer. Okay? This then, too many cyclins, too many CDKs, this will help bypass all the checkpoints in the cell cycle. Now, P53, the tumor suppressor gene, again, damaged DNA or DNA that's become mutated, right? It will then code for different amino acids that make up P53. P53 is actually, it's a transcription factor in itself, okay? And what it does is transcription factors, right? They read DNA and they promote kind of the transcription of DNA into RNA, but it is a transcription factor that promotes the production of proteins like one called P21. And so what P21 does is it produces stop signals for the cell cycle. It stops those cyclins and CDKs. In essence, these things are things like phosphatases that remove the phosphate groups from your cyclins and CDKs, stopping them in the phosphorylation cascade. Okay. Also, this transcription factor P53 is also involved in proteins that help with cell repair and DNA repair, and also with proteins that assist the process of apoptosis as well. And then getting into asexual reproduction. So that's all about meiosis and getting into heredity. So I'm going to leave that there for today. I think that's long enough. That was the material that I wanted to cover in class anyways, but we needed to talk about the actual, what the College Board was saying about the exam and how you can be prepared for the exam. So I hope you've done those things. Remember, you need to complete these slides up to the end of, I believe, 140 by Saturday. Good luck. Let me know. Remember as well, we're going to try and get through the end of this, but next Friday, you're going to have your test. So there's no way we can get through all of this by next Friday. Okay? We still have heredity to look at. Luckily, heredity is not that bad of a subject with Mendel, and you should be able to identify different genetic diseases, all that sort of stuff. Maybe what I'll do is, now that I think about it, we'll finish talking about meiosis, but then maybe we'll go into DNA and gene regulation, because that's more of a meaty topic that we should look at if we have time, because I think a lot of us did pretty well with heredity because it tended to be a little bit more mathematical. Okay, good luck. See you soon.